Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia. Today we are going to discuss about the module binding parameters of ligand polymer complex, Scatchard plot and neighborhood exclusion method from the paper technique used in molecular biophysics part 2. After this lecture, you will be able to understand basic concept of ligand receptor interaction. Then we talks about the forces that involved in the protein ligand interactions. Then we talk about the protein ligand binding kinetics. Then Scatchard plot and neighbor exclusion model in detail. So let's first discuss the protein ligand interaction. So protein ligand interaction is an essential process for the biological macromolecules. They are able to interact with the various small and large molecules with high degree of specificity and with high affinity. And still this process is a fascinating for the biologist as well as the modern biochemist. Protein ligand binding has an important role in the function of living system and is one of the methods that the cell uses to interact with the wide range of molecules. Binding occurs at a particular site in protein molecules, and the understanding of molecular recognition process of a small ligand and biological macromolecules require a complete characterization of binding energetics, and correlation of thermodynamic data with the interacting structures are involved. A close insight into the binding process is of significance and practical interest, since it provides a fundamental to understand the structure-based drug design development strategies. Here we discuss the types of biological interactions. Biological interactions are of many types depending upon what kind of biological molecules are interacting. The first one is the protein interaction. Here the one protein can bind with the other protein and many other proteins. So the protein interaction is totally based on the surface contact and the shape complementarity, and based on that these complex are stabilized by van der Waals interactions, hydrogen bonding, and some of the electrostatic forces. The other type of interaction in the biological molecules are protein DNA interaction or protein RNA interaction. DNA recognition by protein is primarily mediated by certain class of DNA binding domain and motifs which specifically recognizes particular sequences of DNA. On the other hand, the RNA recognition by protein is primarily mediated by certain classes of RNA binding proteins that having RNA binding motif or the domains. So the changes in the structure of DNA that can be induced by protein, through such mechanism the protein regulates gene expression, repair of DNA, and carry out other functions. Moreover, the protein ligand interaction is useful for the drug designing, where a drug interferes with the function of disease protein by binding to it. This interference stops the disease process, and this is the main tool utilized for the structure-based drug design. Here you can see in the figure that the protein have certain binding region where a particular ligand can easily bind in the cavity, and the complex is stabilized by a large number of non-covalent interactions. Here I try to show some type of protein ligand interactions. You can see here the ligand are the small molecule which can bind to the specific binding site present on the receptor and stabilized by varieties of non-covalent forces, including hydrogen bond, electrostatic interaction, van der Waals interactions. So the ligand binding, the ligand can bind to the cavity of a protein, and that can either activate or inhibits the corresponding biological functions. Protein ligand binding is an spontaneous process, and similar forces as in protein folding are work. Function of protein is defined through its interaction and other biological molecules. So the protein ligand interaction is controlling a large number of biological phenomena through the proper interaction that leads to the certain activities further. Now we discuss about the protein ligand interaction energy. The energy is needed to make the things or objects to change, such as movement, chemical reactions, binding, dissociation, and structural changes, and conformation changes as well. So in all these process, a lot of energy involved. That's why it is necessary to understand the complex of energy involved in the protein ligand interactions. It is used for the understanding of binding of the ligand, which induces some structural changes and conformation changes in the receptor that to bind strongly with the described or the given receptor. So protein motion is another thing which required some sort of energy, because the internal motion of the protein has needs some sort of energy to force to make it the different states such as on and off state. A ligand with sufficiently low ligand protein potential energy is considered as a good molecule for the drug design and development. Now we talk about the enthalpic and entropic contribution in the binding free energy. You know that the non-covalent association between two molecules occurs only when it is found to be associated with the negative Gibbs free energy of the binding, and the sum of the enthalpy and entropy is given as a particular equation, that is delta G equals to delta H minus T delta S, where delta H is the enthalpy and delta S is the change in entropy. The enthalpic contribution to the free energy gives the measure of strength and specificity of the interaction between the protein and ligand. However, the entropic contribution in the process illustrates whole dynamics and the overall of dynamics of the overall system, that how the ligands search for the best conformation to fit themselves in the particular receptor. The Gibbs free energy equation can further explain in the other way, that is the delta G equals to minus RT ln Kd, where Kd denotes the binding constant and R represents gas constant and T is the temperature. Here we discuss about the Gibbs free energy. The Gibbs free energy is the measure of the protein ligand interaction, and you know that you can see here for the endogenic reaction delta G is more having the positive value. So the work done for raising object, if the delta it is presented in a way that the for the delta G is negative less than 0, means it is the exogenic reaction where the loss of potential energy as the position. So the delta G can be defined in terms of enthalpy and entropy, and it is equals to the delta H of the system, enthalpy of the system, minus T delta S of the system. For a reaction to be spontaneous, the entropy of the universe delta S total must increase. So the delta S is, if the delta S is more than delta H by T, or delta G equals to T delta H system minus T delta S system is less than 0. So the free energy must be negative for a reaction to be spontaneous. Here we try to explain the Gibbs free energy. Again, the Gibbs free energy depend on the either on the enthalpy or on the entropy. So their both terms are shown here. For you can see here for the enthalpy, you see that the things want to roll down the hill. So delta H tends to be negative, and it is the dependent on van der Waals interactions, hydrogen bonding, and charge-charge interactions. However, the entropy mediated system, thermal or Brownian motion tend to keep things uphill. Delta S tends to be positive. So it changes the arrangement of solvent or counter ions. It reflects the degree of freedom and rotational and translational state. So there are 4 cases in which the delta G either positive or negative. The first case, the delta G is positive because the delta H is positive and the delta value of delta S is less than the delta H. So the overall value of the delta G is positive, while in other case the delta H is negative and delta S is also negative, less negative, then ultimately the value of delta G is negative for the spontaneous process. In the third case, the delta G is again positive because the value of delta J H is positive and the value of delta S is negative, so ultimately the value of delta G is positive. And in the fourth case, the value of delta H is positive and the value of delta T delta S is more negative, so the overall value of delta G is negative. Here we try to explain the binding free energy in terms of interaction of the ligand and protein. You can see here in terms of delta G, the receptors, the ligands bind completely into the cavity of the receptor, and the quantitative description of protein ligand interaction can be given as protein plus ligand gives rise to PL complex, that is protein ligand complex. So the association constant is simply Ka equals to product by reactant, means concentration of PL complex divided by concentration of P and concentration of L. So the fraction of the ligand bound and the unbound can be further explained in terms of theta, and that's equals to PL divided by PL plus P, means the binding site occupied divided by total binding site. After rearranging this equation, you can get the value of theta as a ligand concentration divided by ligand plus 1 by Ka. Here we describe the quantitative description of protein ligand reaction. So there are many types of the interaction, multi-ligand interaction. They have more than one binding sites. You can see here there are binding sites for ligand A, so Kd value for the A, Kd value for the B, and Kd value for the C are the different. And the value of theta is again represented in terms of ligand plus ligand divided by ligand plus 1 by Ka. After rearranging this equation, since Kd equals to 1 by Ka, you will get the value of theta L ligand concentration plus ligand concentration plus Kd. So this is plotted in the form of a graph, and you can see the value of Kd where the 50% of the ligands bound to the protein. Here we discuss the types of forces that stabilized protein ligand interactions. So you know that there are 5 different types of interaction that helps to stabilize the protein ligand complexes. These forces are represented here in the way which are formed. So your first one is the hydrogen bond, which is formed between the OH group and with the oxygen hydrogen bond donor and acceptor pairs. Others are the hydrophobic interaction, where the two hydrophobic molecules are together, can be able to form the van der Waals interactions. The third one is the disulfide bond, and the fourth one is the ion pair. Apart from that, the cation pi-pi interaction or aromatic ring stacking is also participates in the formation of protein ligand interactions. Now talk about the charge-charge interaction. The Coulombic interaction between two ions at close range are very strong, as good as the covalent bond. Their energy decreases with the inverse of the radius and falls off less than kT at a power about 56 nanometer separation between the two charges. In practice, charge-charge interaction have been shown to be chemically significant up to 15 angstrom in protein. A small charged metal ions can act as a positive charge in an ion pair. You can see here in the diagram that how the charge-charge interaction takes place in a protein molecule. Let's explain the hydrogen bonds. You can see here the hydrogen bond can be formed only when the hydrogen is attached to more electronegative elements such as oxygen, nitrogen, and fluorine. So you can see the non-covalent chemical bonds. The hydrogen bond is a non-covalent chemical bond in which electronegative atom, a hydrogen bond acceptor, shares hydrogen atom with an electronegative element with a bound hydrogen. The energy is 10 to 40 kilojoule per mole, and its distance is around 1.7 to 3 angstrom in the length. The strength of hydrogen bond varies with the angle of the hydrogen bonded interaction as well as the length, of course. Individually not very strong, but the large number of hydrogen bond in regular secondary structure stabilize the framework of the protein. Let's discuss about the van der Waals forces. You can see here that the van der Waals forces, also known as London forces, it is a weak type of interaction caused by the momentary changes in electron density in a molecule. They are the only attractive forces present in the non-polar compounds. So this is the non-specific forces between the like or unlike atom, and it decreases with the r to the power 6, and its bond energy is approximately 1 kilojoule per mole. And if the r naught is the sum of the radius van der Waals radii of the two atoms, van der Waals forces are attractive forces if r is more than r naught and repulsive if r is less than r naught. So it is a distance dependent force. This is the weak interaction, and temporarily dipole constitute the van der Waals forces. The surface area of the molecule determines the strength of van der Waals interactions between the molecules. The larger the surface area, larger the attractive forces between the two molecules, and the stronger the intermolecular forces. Here we describe how to calculate the protein ligand binding affinity. The dynamic interaction between protein, defined as target here T, and ligand L assumes the simple in equilibrium. L plus T gives us LT complex. So in the K, if the reaction is K on, means the LT complexation of LT occurs. In the K off, dissociation of LT occurs. So the equilibrium of binding constant is defined as Kb is simply the K on by K off, and the equilibrium terms are given at the numerator and denominator accordingly. Similarly, the dissociation constant is defined as the K off by K on, where LT at equilibrium, T equilibrium, and L equilibrium are the equilibrium concentration of the complex, free protein, and free ligand respectively. So it is obvious that Kb is inverse of Kd. Now we discuss about the equilibrium constant measurement methods. There are a large number of methods available to determine the equilibrium constant or binding affinity between protein or receptor with the ligands. The most common and simple method is the fluorescence spectroscopy. The fluorescence spectroscopy is based on the either in the fluorescence and isotropy, which have a limitation of a small fluorophore, and the other is the fluorescence absorbance spectrum shift because of the change in the microenvironment of the system, which causes the change in the lambda max from the either to the blue shift or the red shift. The another interesting method is the capillary electrophoresis. The capillary electrophoresis affinity electrophoresis is the non-equilibrium capillary electrophoresis for the equilibrium mixture. The third one is the surface plasmon resonance, which is based on the response unit and the binding of the ligand to the coated gold plates. And the fourth is the isothermal calorimetry method. This is the based in the solution system. There you do not require any support, and this is the most extensive used method for the measurement of binding affinity between the protein and ligands. Now we explain the principle of isothermal calorimetry. Isothermal calorimetry method is isothermal titration calorimetry, simply known as ITC, is based on the heat generated or absorbed in every chemical process. This technique is implemented in the solution form. So both protein and ligand can be taken in the solution, and it is a real-time and direct measurements of the protein-ligand interactions. There is no limitation of molecular mass of the ligand or the protein. There is no labeling required, and of course there is no any optical limitation. ITC is a quick and accurate measurement of binding affinities. The mechanism of action and conformation changes occurs based on the mechanism of action and conformation changes, and of course a structure and function relationship can be established between protein and ligand, and you can differentiate between the specific versus non-specific binding in the ITC. Through ITC, we can measure the binding affinity, the mechanism of the binding, number of binding site, and kinetics of the reaction. Here we discuss again the single experiment, ITC as a single experiment, which is sufficient to obtain all the thermodynamic components. The meaning of ITC data is simply if for a exothermic reaction, the negative peak should be observed in ITC. For the endothermic reaction, a positive peak is observed on the ITC, and the heat absorbed or generated during the titration is directly proportional to the amount of bind ligand. So based on that, we can be able to understand the mechanism, we can calculate the binding affinity, and we can calculate the number of binding sites present in a particular receptor for the given ligands. Another important technique for the estimation of binding affinity between the protein ligand is the surface plasmon resonance, known as SPR. SPR is an resonance between light and a cloud of free electrons in thin layer of gold. Electromagnetic field of light penetrates inside the gold to a small depth, and resonance results in the dissipation of light energy and thus decreasing the intensity of light reflected from the gold surface. So you can see here the sensogram of the SPR, where the time is at the x-axis and the response unit is at the y-axis. And if you get the plot when there is no response once only buffer is flowed, but once the sample is injected and if association occurs, you will get an immediate increase in the response, and the time for the steady state is showing the how strongly this complexation occurs. Then the another dissociation buffer is passed and you can see the complex is dissociated, and then further the things are regenerated. So the value of K on versus K off is taken to estimate the value of Kd in the SPR. So when the protein bind to ligand, the angle of minimum reflected light changes, and change is proportional to the mass of protein bound to the surface. So this is one of the most efficient technique used to measure the binding affinity between the protein and ligand. Here we define the Scatchard plot. It is a simpler model of interaction which was proposed by Scatchard in 1949. This model works well for small ligands which occupy only one binding site of a one-dimensional lattice. The interaction process can be summarized by the chemical reaction. For example, CF plus CFS give us to CB, where CF is the molar concentration of free ligand in solution, CFS is the free binding sites, and CB is the bound molecules. The parameters K1 and K2 are the association and dissociation rate constants. At chemical equilibrium, we can write CB divided by CF into CFS, that's equals to K1 by K2 or equals to Ki, where Ki is the intrinsic binding constant. A Scatchard plot is a graphical method has been used to analyze the ligand binding data at equilibrium with a particular ligand concentration, free ligand concentration, and average number of molecules that can be bound to receptor are the experimental parameters used for the Scatchard plot, and N can be defined as moles bound ligand divided by moles of the receptor, that is the sum of the bound ligand divided by sum of the receptors. So in the simplest case where the receptor with only one single class of non-interacting sites is present, the Scatchard equation for the receptor ligand binding will be V by L equals to NKa plus VKa. By using logarithmic scale for x-axis as shown below, for the same binding data, the data get more spread out at low value of A and form a characteristic sigmoidal curve. The best results are obtained if binding data over several order of magnitude in A concentration can be collected. So once we can linearize binding data using so-called Scatchard plot, starting with the equation V equals to Ka into concentration of A plus 1 into Ka into concentration of A. By multiplying both sides, 1 plus Ka into A yields V plus VKA equals to Ka. Dividing through by A gives the V by A plus VKA plus Ka, which rearranging in the terms of V by A equals to Ka minus Kav. So by now this you get the linear equation. So calculate the binding affinity. Here we try to show the saturation curve and Scatchard plot. Ligand bind to the protein, you will get this kind of curve where Kd can be estimated at the 50% of bound and free ligands. But by rearranging the same thing in the other using the Scatchard equation, you will get linear graph, and the value of slope is equals to the value of Kd. Here we try to show the Scatchard plot for the more than one binding or n number of binding sites. For multiple binding sites, n identical and independent. If instead we have multiple identical binding sites, P plus Na equals to PAN, then it is possible to show the appropriate form of the Scatchard plot is V by A plus Nka minus Kav. A plot of V by A versus V should be given a straight line for the binding reaction with identical sites. The slope of plot minus Ka and the Y is the intercept at Nka. For n sites on the macromolecules, concentration of bound ligands Pa plus 2Pa2 plus 3Pa3 and so on. So it is written as sum of the I plus I and PAI, and I limit of I is 1 to n. So concentration of macromolecules equals to Pa0 plus Pa plus Pa2 plus Pa3 and so on. So the value of V can be obtained as a sum of the PAI divided by sum of the PI from 1 to I equals to 0 and I equals to 1 to n. Here we try to show the different types of binding pattern. For example, if you in the first curve you can see the two different binding sites and behaves differently. In the second term, you can see the multiple binding sites for the polyclonal antibody, and in the third one, you can see the reaction of cooperativity. Here we present the Scatchard plot for a receptor of 3 equivalent non-interacting ligand binding sites, and the slope is equals to Ka. And the second is the example illustrate a molecule show the Monod-Weinmann-Changos concerted model of allostery with 6 binding sites and 10-fold preferential binding to the R state. And the third one is the multiphagic Scatchard plot for the N1K1 plus N2K2. Now talk about the Neighbour-exclusion model. The Neighbour-exclusion model is a classical model used to explain DNA interaction with the ligand molecule. This is the graphical behavior of the Neighbour-exclusion method shown here. The closed form of this model was formulated in 1974 by McGee and von Hippel and even used to analyze the experimental data these days. This model can be used to study the variation of the contour length and the variation in the elasticity of DNA ligand complex. At saturation, this would give rise to a binding stoichiometry of one base pair per bound intercalator. So now summarize this module. So protein ligand complex having a strong entropy contribution tend to have weak enthalpy contribution, and the complexes with very strong enthalpy contribution found to have weak entropic component. We have described in detail about the enthalpic and entropic component of the binding free energy. Then molecular recognition provides a strong mechanism for exogenous control of biological system, and many medications and biological probes performed by binding and usually inhibiting a precise macromolecular target, which is extensively used for the drug design and discovery component. In fact, the major Precautions of the early stage drug discovery is the screening of a ligand that bind specifically to target protein with high affinity. By the Scatchard graph, we can calculate the number of ligand binding site present in the receptor. The determination is irrespective of cooperative interactions, number of class of sites and their affinities. Thank you.